Let's walk through a spatial transcriptomics analysis in Illumina Connected Multiomics. We're going to start from our project and add a new analysis, name it accordingly, and select the analysis type. In this case, we're going to be looking at default spatial transcriptomics. We have a sample already added to our project, so we're going to select that and run the analysis. Once that's complete, it's going to appear in our overview page. Clicking on it, it's going to open our spatial report. This opens in the data viewer, and it's going to allow us to visualize the initial results from our DRAGON spatial transcriptomics secondary analysis stage. We have a UMAP, we have the Leiden clusters that have been generated, and we have the spatial analysis with that information overlaid on top. Here we are visualizing one of the Leiden clusters, which resolves an anatomical feature in our sample. We can switch between any of these clusters to visualize them better. Zooming in, and we can look at the cell boundaries and nucleus boundaries in our spatial image. The style menu allows us to, for example, change the opacity of those cells to better view the underlying image and anatomical structures that we are observing. We can change that and revert the change as needed. We can also use our tools in the data viewer to select specific groups of cells within the image. We can use this for later classification or for filtering out cells that we're not interested in analyzing. We can do this at the cell level as well as the whole region level in our image. Once we're happy, we'll save this data viewer session for later use. We can now go back to our spatial analysis tab, which takes us to the analysis tab in our project. This is going to be the place where we can repeat the analysis, look at any of our results, as well as doing further analysis. Here I'm clicking on Rerun task on the DRAGON spatial transcriptome, which was previously generated. This is an all-in-one task which allows us to do cell filtering, normalization, feature selection based on variability, as well as computing the UMAP and Leiden clustering. As you can see, I'm changing some of the parameters. This can be repeated as many times as needed until we generate some satisfactory results. As you can see here, by clicking Finish, I'm generating a new branch of analysis in my project. So you can have as many of these as needed. The DRAGON spatial transcriptome task generates two nodes, one which contains the clustering results and one which contains our filtered results, which we are going to be using to classify our cells. Here I've selected the SCAMPI-SC type task for cell classification, and I'm changing the significance threshold as well as the tissue type. We have a mouse brain, so we select the brain, as well as selecting the recompute option for a clustering attribute. I'm changing the clustering resolution to match my previous Leiden, and then I'm clicking Finish to run the task. This is going to generate a new node which contains the single cell classification. I can then use that information to run a compute biomarkers task. This is going to compute the differentially expressed genes between the levels of our attribute. We can choose the attribute from the drop-down menu. In this case, I'm looking at the SC-type classification, and I'm choosing to only look at upregulated genes, so the positive biomarkers for each one of our cell types. Having computed my biomarkers, I can now go back to the data viewer to visualize the results from our cell classification. Clicking on the spatial report will open a data viewer session, similar to what we've had before, but I'm now going to change the information visualized. I'm selecting the node from our style menu and selecting the SC-type classification attribute, which will now be visualized on the spatial image. You can transfer the information across plots in the data viewer by simply dragging and dropping that information. You can also use the axis menu to select the information that is being visualized. Again, here I am changing the information on the spatial image, the UMAP, and the pie chart to visualize my SC-type classification results. I'm also going to add a table at the bottom of my spatial image to look at the biomarkers that I have identified for each one of the cell types produced by the SC-type classification task. Again, by clicking on the pie chart, I can highlight any of those classifications on the image. I'm zooming in right now to visualize the radioglial cells that we have identified thanks to our classification task. Again, I will change the opacity of these cells with the Style menu option. This can help me focus on just the cells that I'm interested in, as well as looking at the underlying anatomical structures in my image. It is sometimes interesting to look at specific biomarkers for these cell types. So I've duplicated my spatial image, and then I will be dragging and dropping one of the biomarkers onto the image to look at the expression of that specific biomarker in the spatial context. So I will be zooming in the same region, and now instead of looking at the classification, I'll be looking at the expression of one specific gene in those clusters. By reducing the opacity of those cells, I can at the same time look at the underlying structures in my samples, or I can focus more closely on the expression of that marker in each one of those specific cells. Working like this in the data viewer can allow you to very quickly assess your data quality and the quality of the biomarkers that you have identified. Again, we're now saving this session for later use or later revisitation, and we'll be naming it cell classification. Having run sctype, we'll have an attribute for the classifications, but that will be only available in the single cell node. We'll use the publish cell attributes to project task to make that attribute, the sctype classification you're seeing on screen, available to the entire project. We'll have to rename the attribute so that it exists uniquely at the project level. Once you're happy with the name, simply click Finish, and the attributes will be made available to the project. Now that we have identified our cell types and computed biomarkers for them, we can normalize our data. We'll start from the filtered node and use the recommended methods for the normalization task. This will be then used to run some differential expression analysis between cell types that we have identified. The differential expression analysis tools are found in our statistics menu under the differential analysis task. You can choose between a number of methods, but for this we will recommend Wilcoxon. This is the SCAMPI implementation. Clicking Next and then selecting the correct factor for the analysis, in this case the sctype classification, will allow us to set our comparison. We'll be looking at the difference between the radial glial cells that we just visualized, and we will be comparing those against all the other cell types that we have identified. You could select one or any combination of those. Once you're happy, click Add Comparison and click Finish to run the task. The differential expression analysis task will generate a report which we can access by double-clicking the new node. This report contains our p-values and corrected p-values, as well as the fold changes for all the comparisons that we've run. We can filter this based on, for example, the corrected p-value from the menu on the left-hand side. Here I'm setting up a significance threshold of 0.05. This automatically updates the list of genes in our report, filtering down to about 2,000 genes in this case. We could also use the fold change for this significance threshold. By clicking the Generate Filtered Node, this will queue up a task that generates a new filtered node for later use. We can also access a report in the data viewer, which creates a volcano plot, a list of genes, as well as the dot plots for some of the most highly significantly differentially expressed genes. I am expanding the volcano plot and changing the size of the points using the Style menu for better visualization. And by clicking on one of those points, I can also visualize the name of the specific gene. We could use the data viewer for filtering, but as we've already done it in the report, there is no need for it. We'll save this session again for later use or for later reporting of our results. Now that we have our list of differentially expressed genes, we'll be using that to run a gene set enrichment task. This is found under biological interpretation. For this specific project, I'm using the KEGG database, and I will be using the mouse database. Clicking Finish will again run the task and generate a report. We can access that report by double-clicking on the node, and this has all of our pathways and the level of enrichment, including the enrichment value and the corrected p-value. By clicking on any of the pathways, we can visualize the pathway directly, as well as the genes that are differentially expressed within that pathway. We can also filter for significance, in this case, the enrichment score, and this will let us automatically open the plots in the data viewer. We can now generate a number of plots, which are going to summarize our results for this analysis. Starting with the spatial plot, we will select our initial single cell node, and then as it loads, we will select the x-axis and y-axis data with the spatial x and spatial y data. These are the spatial coordinates for each one of our cells. We will then go ahead and add a pie chart, much like before, recreating our initial spatial report. We will select the SC type classification for this, and we will also add a volcano plot with the results of our differential expression analysis. This gives us a general overview, but we can see that we have a number of NAs in our pie chart. These are all the cells that were filtered by our initial analysis with the spatial drag and transcriptome task. We will be filtering these out as we don't have any results for them. We can do that by going into the Select and Filter menu, adding the SC type classification as a criterion, and then deselecting NAs and including just the cells that we do have a classification for in our analysis. Again, clicking on the pie chart lets us easily visualize the classification for cells that we do have in our analysis. We're going to go ahead and add a new plot. It's going to be a table of biomarkers. These are the ones that we have identified with the Compute Biomarkers task for the classifications of our cells. We can also rearrange any of these plots as I'm showing you right now. We're going to put the volcano plot on the right and then rearrange the table of biomarkers to the bottom. This is going to give us a better spacing for our plots. Finally, we're going to want to visualize some of the expression of these biomarkers, and so we're going to add a further plot, a 2D scatter plot. This is going to be a dot plot showing the expression of those biomarkers in our different cell types. We're going to look at the normalized expression data because it's going to give us a better visualization. For the x-axis, I'm going to choose the SC type classification. This is going to split our data in each one of the levels and types of classifications that we have for this project. And then I am selecting one of the markers to show the expression of on the y-axis. Our style menu lets us customize our plot. We're going to color by classification. We are also going to add a box and whiskers to that plot. We're going to overlay it and color it for better visualization. And as I expand that plot, you're going to see the visualization of the expression of that specific marker in our cells. So we can see that this radial glial cells marker is mostly expressed in that cell type, but the box and whiskers plot really helps us clarify the levels of expression. And you can of course switch between markers as easily as that by dragging and dropping other markers on the y-axis. So this gives us a summarization of all the results that we have generated, and we are going to go ahead and save this for later. You can save this for presentations as well as revisiting this in the data viewer later on, and we're going to name it Results. Saving this, we're going to go back to our Analysis tab to look at the pipeline that we have generated for the analysis of this project. We have gone from our raw data, we have repeated some of the results that we had generated in the secondary analysis, and we have then classified our cells, generated biomarkers, normalized our data, and run differential expression analysis. Based on the results of the differential expression analysis, we have filtered significant genes to then use for the gene set enrichment and biological interpretation or our analysis. We have then summarized all of our data in a data viewer session showing those results. This should give you an end-to-end overview of spatial analysis in Illumina Connected Multiomics.